All right, welcome everybody to the Connect Science series here at Dana Farber Cancer Institute. I'm Rafael Rizari. I'm the chair of the Department of Data Science, and I'm hosting one of our data science talks in this series. We're very fortunate to have Rafael Gotardo joining us today. Rafael is a scientific director of Translational Data Science Integrated Research Center at Fred Hutch. He's a professor at the Hutch as well, and at the Public Health Science Division there too. He is the J. Orrin Edson Foundation Endowed Chair, and I've been following Rafael's work for, wow, I don't want to say how many years now because then we'll feel old, but it's more than 10. And I first learned of his work when he was working on flow cytometry, and now more recently on single-cell RNA-seq. He is, like many of the people in our department, he's very interested in applications and in advancing understanding of cancer. So he's a very applied statistician, as I'm sure you're gonna see today. So thanks again for joining us, Rafael, and we look forward to your talk.
Thanks, Rafa. So yeah, it's my pleasure to do this. And what I wanted to do is to tell you about some very recent work that we've been doing on sort of analyzing spatial transcriptomic data. Before sort of jumping into this, I'll give you some background into some of the work we've been doing, while we're trying to do it specifically within the context of Merkel cell carcinoma. So my lab has some conflict of interest. We collaborate a lot with biotech and pharma on various projects. So in general, what we're trying to do is to really leverage data science to better understand and ideally improve clinical response. And in my lab, we've been doing lots of work on sort of immune profiling, looking at vaccine infectious disease, but also looking at immunotherapy. And so here I'm giving you two examples, looking at anti-PD-1 response in the context of melanoma, or looking at vaccine efficacy for the RV144, which was the first HIV vaccine to show some efficacy in humans. And so ideally, what we want to know is what's really different between the people who respond and have a durable response, or in the context of the vaccine, what's different between the people who potentially are protected or have a decreased risk of infection after vaccination. So, we've been lucky that things have improved a lot. Sorry, I skipped the slide in terms of what we can do for immune profiling. So ideally, what we'd like to do is we're seeing, volunteers or patients, as part of a clinical studies, and we want to do sort of longitudinal sampling, looking at, maybe blood, maybe sort of tumor biopsies, and using these sort of new high throughput technologies, including maybe multi-dimensional flow cytometry, single-cell RNA-seq, and ideally even spatial profiling on some of the tissues. We want to do that before they receive treatment, right after, and maybe long-term follow-up to look at, what's the long-term consequences of some of these treatment or immunotherapies. And then correlate some of these data with the clinical response, whether we're looking at just, the response, maybe adverse events and side effects. And things have changed a lot. So this is sort of, giving you a snapshot of what we used to be able to do. This is one of the first flow cytometer where you could measure basically 1 to 2 proteins. At the same time, doing computing was very difficult. You needed to have access to these sort of, supercomputers. But today we have, amazing technologies, high dimensional, single cell for the most part, either looking at sort of genomics or next generation flow cytometry, or even spatial transcriptomics. At the same time, we have computing on demand, so we can do things in the cloud very efficiently. We can really scale as we need, so there's not much limitation from that point of view. At the same time, there's been a lot of improvement in terms of the data that are available. This is actually showing you, a snapshot of the Human Cell Atlas in Immune Space, which is a database as part of the Human Immunology Project Consortium that I'm leading. And then we have really, a lot of advances in terms of computing and algorithms that we can use, thinking about AI and deep learning, but also Bayesian modeling and other things that have really benefited from the data that are available and the computing that has increased significantly. Okay, so let me bring that back to why do we need all of this? Why is this important, right? Sort of thinking about what Rafa said is really thinking as an applied statistician is what can we do to help in terms of understanding responses to, let's say immunotherapy in our case. So this is the case of Merkel cell carcinoma. So for those of you who don't really know, this is a skin cancer. It's actually pretty rare, but it's increasing in the US. It's actually very aggressive. It's twice as likely to metastasize as melanoma. What's interesting about Merkel cell is actually at least in the US, 80% of the cases are caused by a virus. So if you think of immunotherapy, that provides a really great target for going after the cancer. Unfortunately, up until recently, it was very difficult to treat. All you could do was delay the progression of the cancer through standard chemotherapy. So we've been lucky to collaborate with experts here, including Paul Niem at the FedEx and the University of Washington. And so we've got a P1 to actually do that and really understand, the concept of response or lack of response after immunotherapy. So just to sort of, again, motivate what we're trying to do, I told you up until recently, there was basically no treatment. It turned out that anti-PD-1 treatment was shown to be effective through a clinical trial called CITN-09, which was partially run at the Hutch. And so this was great because we've shown that just like in the context of melanoma, with Merkel cell, anti-PD-1 therapy could really improve sort of responses to this treatment. And actually, in some cases, you can have very durable response. However, if you look at the FDA announcement, you will see that, great, the overall response rate was around 56%, which is good given the fact that there was nothing. But then if you look at the complete response rate, it's only 25%, right? So it's good, but it's certainly not good enough from the point of view of our patients. And so here I'm giving you a little bit of background on some of the work we've been doing before jumping into where we're going next with the spatial profiling. One of the things that we were really interested in, so, as Rafa said, my group has done lots of work on sort of high dimensional flow cytometry data analysis, and we've been doing this for over 10 years. And so we've always been interested in using these kinds of data because they're great. They're, really high throughput. You get millions of cells, and because they're still sort of cheap and affordable, at least compared to sequencing, you can profile hundreds of samples potentially. So we get access to these sort of larger cohorts when you compare that to single cell genomics. So we actually had access to some of the data that were generated as part of the CITN-09 clinical trial. So they had actually run a T cell panel, so trying to understand the state of the T cells and before and after treatment in this trial for anti-PD-1 in the context of Merkel cell. There were 27 patients, 18 of which responded to therapy, so the responders, and then basically you had sort of 9 non-responders. They had run a pretty standard T cell panel looking at basically, 15 or so markers, some, delineate used to find your standard sort of phenotypes, such as your CD4s and your CD8 T cells. And then you have your sort of activation, exhaustion markers and things like that. So one of the things that we wanted to do, and we had built an approach to do that, a new computational methods to sort of identify phenotypes in an unbiased manner for flow cytometry. So we wanted to identify baseline T cell predictors of responses to anti-PD-1 using this new approach that we call FAST. So we have actually a preprint that's available in bioRxiv. So I'm not going to tell you too much about the approach, but just to give you some background on why this is important, what we can do when we really think about new computational methods for analyzing these kinds of data, which in this case are not necessarily super new. So we actually went ahead and analyzed this data. And what we were told at the time is, yeah, we're happy to collaborate with you. Here are all the data sets, but just to let you know, we've looked at it and there was nothing that was available. So this being said, we still went ahead and analyzed this data, and only looking at the baseline measurements. What we found are sort of 4 baseline predictors of responses to immunotherapy. Basically, there was 2 that were CD4s and 2 that were CD8 sort of T cell subsets. Here, what I'm showing you here is the frequency of these T cell subset broken down by responders and non-responder status. So on the left-hand side here, what you see are the non-responders. So this is the frequency of these T cells that we're finding at baseline in the blood. And you can see that the responders have a higher frequency of these T cells that we're finding. If you look at the phenotype that is shown at the top, it's actually showing you that these T cells are PD-1 positive. there's actually a dim and a bright expression of PD-1 that we're able to find. And that kind of makes sense because this is an anti-PD-1 therapy that is basically trying to block PD-1s, releasing the brake on these T cells so that they can really go after the T cells. However, it's not just PD-1 positive alone. What we've identified is a combination of HLA-DR, PD-1, and CD28. CD28 has been shown to be actually critical for anti-PD-1 response. So this also made sense, but our collaborators never really look at this combination because you have to think about the fact that this is done manually when they look at combinations, and there's an exponential numbers of things that you have to look at. So again, this is just to show you that by using new approaches, new computational approach, we were able to identify these predictors that made a lot of sense given the biology. And we've shown actually that these also correlate with what we're seeing in the tumor, such as anti-PD-1 staining, or TCR clonality. So we have other additional data that's shown that these sort of made a lot of sense given the other data that we had. In addition to that, we actually went back, and I'm not going to show you that, but we went back and used some of the frozen PBMC samples that we had and validated these signatures further on new samples. However, we wanted to also see whether we could sort of look at additional data sets that had been sort of published in the literature in terms of signatures, maybe looking at other cancers, and specifically we looked at melanoma. There was actually a data set that was published, or a study for which the data set was available. This was actually a CyTOF study, but fortunately for us, they actually, all the markers we had were a subset of some of the markers they had in their panel. So we reanalyzed this data set looking at the same markers, and again, even though the significance is not as high as our study, you can still see a segregation or a difference between the responders and non-responders. Different cancer, melanoma, different technology. Here's CyTOF versus cytometry. Different sample types. This is frozen PBMCs. In our case, was done on whole blood, fresh, but still we can sort of see that the trend actually holds in this data set. Okay, so that's story number one, just to give you some background into what we're doing in the context of Merkel cell. The other one that we've done is, this is actually looking at a slightly more complicated immunotherapy. This is a TCR-based cellular immunotherapy, and this was actually combined to anti-PD-1. So this is actually a specific patient that was seen here in clinic that responded initially very well to T-cell therapy. So this is actually showing you the response here on that graph. This is the disease burden. And what you see is that, the disease progressed even after anti-PD-1 therapy. Then the T cells were infused, and then you can see a really nice response where the tumor is basically almost completely gone, to the point that patient actually went back to work and had a very durable response for over a year, but then eventually, relapsed and eventually died, actually. And so one of the questions that we had is, what happened? How come the patient initially responded very well, but then relapsed? And this is actually the work of Kelly Paulson, who used to be a postdoc in Ochapu's lab in the clinical space, and the work of Valentin, who was a postdoc in my lab. So what we wanted to do is to use single cell genomics to try to understand the lack of response, or at least the progression even after initially responding. So we actually looked at PBMCs, and we also looked at these tumor biopsies. So here I'm only going to show you the data from the tumor biopsies. So we actually had the ability to do single-cell RNA-seq right at the beginning of the study, and once the patient relapsed. And so we actually profile these two tumor biopsies. And again, you have to think about the fact that here we can do that because this is looking at a skin cancer, so we have access to tumor biopsies. So we run, this is actually one of the very first single-cell RNA-seq experiment that was done on tumor biopsies at the Hutch. It's still, it's already, I believe, 2 or 3 years old. So this was using, one of the first technologies in collaboration with 10x. So when we run the two samples, what we realized, this is actually showing you a t-SNP plot where each single cell is a dot, and there's two colors. The blue is the cells that come from the tumor before, and the orange are the cells that come from the tumor after. So when we looked at these data sets, we saw a major shift between the before and after. And so, thinking as a statistician, you're always worried about maybe batch effects and things like that. So we felt, well, is this really normal? Do we see a major shift in the expression profile of the cells in the tumor before and after therapy? However, if you look at the cells that are part of sort of the immune system, such as your TILs or your macrophages, you can see that there's overlap there. So only the tumor cells were really segregated, which made us think that probably was okay. There was good alignment of immune infiltrates, but not of other tumor cells. So we did, the first hypothesis is that, I told you that this was actually an HLA-B targeted therapy. So we looked at the effect of HLA on gene expression. HLA-A, which was not targeted by the T-cell therapy, we saw no shift in the expression of HLA-A. However, if you look at HLA-B, which was targeted by the T-cell therapy, we saw major downregulation of HLA-B. So basically, this shows that the cancer escaped the immunotherapy by downregulating HLA-B, which was not visible through standard IHC or flow cytometry. So this was really nice because we basically saw right away and confirmed one of the hypotheses that was known. This is actually something that would have come as well if you were to do an unbiased analysis of differential gene expression. HLA-B would come up as well. In fact, there's other things that would come up, and we show that also HLA-E was overexpressed, which sort of block NK cell response, which also explain why this patient did not respond very well to NK cell boosting. So this is another example of using single cell technologies to really unravel and understand responses to immunotherapy, or at least relapse in this case. And we're doing a lot of other studies in this context. Okay. So this is sort of the background and introduction, tell you a little bit some of the work that we've been doing in my lab in collaboration with others at the Hutch. So now what we're sort of interested in doing is really trying to understand, the spatial organization of our cells within the tumor microenvironment and its link to clinical response. Still very early on, I'm not going to be able to really tell you a lot of new things today based on that, but at least I'll show you where we're going in terms of the computational modeling. So the thinking, and I'm sure many of you know this and probably much better than I do, is that you can think of it as the tumor microenvironment in these sort of hot and cold tumors, right? The hot is the one where you have the immune cells that are well infiltrated, they are right next to the cancer cell, and they're really acting on the cancer cells. The hot is the one where you've got exclusion, and you've got these sort of pockets of immune cells, but they can't penetrate the tumor. And typically, the cold are linked to poor clinical response, right? Because there's not good immune response on the cancer cells. So what we wanted to do is try to understand the structure spatially, and also look at maybe the new technologies for spatial profiling, and trying to see if we can better understand the immune profile of the cells that are maybe infiltrating versus the one that are not. So maybe different, comparing two different tumors, or maybe within the same tumor, comparing the expression profiles of the T cells or any immune cells that are infiltrating the tumors versus the ones that are not. Okay. So one of the technologies that we've been testing at the Pradesh, it's the Vision Spatial Technology from 10x Genomics. And this is basically a way to do unbiased, high throughput gene expression profiling of tissues. And the idea is pretty simple. Basically, you have a glass slide with these little rectangles there. Each of these rectangles has barcodes on them that represents, that are spatially basically laid out on the slide, and you kind of have that sort of hex representation on the slide. And then what this will do is then this will capture the RNA on each of these locations, and then you can basically get the gene expression, the expression level of all the genes on that spot on your tissue, right? So you can sort of map gene expression on the slide through these barcodes, and then extract the expression levels of all the genes within the given spot. This is not truly a single cell technology yet. It's pretty close to it, and I'll get to that in a minute. But typically, you get about 1 to 10 cells per spot, at least according to 10x. So you do your sample preparation, you put your tissue slice on it, you do imaging, maybe you want to look at the quality or other things, then you can construct your barcode, library construction, you sequence, and then you get all the genes, for a given spot as part of the tissue. So one of the things that we wanted to do is can we come up with new statistical methodologies for analyzing these kinds of data, right? Thinking about really coming from the field of statistics, thinking that now we're looking at these spatially. We know that things will vary spatially on the slide and therefore as part of the tissue. And so given my background, we wanted to look at Markov random fields, which are one tool for analyzing spatial data like this, especially when they are arranged on a lattice like this. By that, I mean that either they're arranged like this, what I showed you. So you have these spots over here, and you can kind of see they are nicely laid out, almost like hexagons. And there's another technology which is called spatial transcriptomics. This is actually the earliest version that was that made it into the Visium platform. This was that company in Sweden, Spatial Transcriptomics, that was acquired by 10x Genomics, and then they've improved it. They've improved the resolution, and now the layout is slightly different. So the old technology had some sort of a more square lattice, and this one has that sort of hex like lattice, like a honeycomb. So I'll tell you a little bit about what we're doing, but basically the key aspect is we wanted to try to use the spatial or the gene expression data to try to identify spot types. And I don't say cell types because remember that this is not truly single cell resolution yet. In fact, I think I have the resolution here. You have basically these spots are around 55 micron, and there's about sort of 100 micron between the center of two spots right next to one another. So 1 to 10 cells per spot. So we'll talk about sort of spot types as opposed to cell type. So what we wanted to do is to, again, identify the different kinds of spots that we have within the tissue, and then maybe try to map those into what we know about maybe, cancer or immune cell types so that we can better understand the architecture of the tissue. So one of the things that we wanted to use is what we call a pot spatial prior. I'm not going to go into the details, but basically, if you think about different spot types, here we have sort of different colors. And what our prior is going to say is, I want spot types or spot of the same types to be that are more likely to be right next to one another. So what this prior says, which is written at the bottom here, it's actually encouraging spot types, spots of the same types to be right next to one another. So if I look at this sort of square lattice here, and if I know sort of these four neighbors here, then the one in the middle, if I don't know the spot type, it's more likely to be blue because there's more blue around. That's what the prior is basically saying, right? Same on the right-hand side, it's more likely to be blue, right? So if I had to make a guess as to what the spot type would be, it should be blue. That's just a prior, so that's just sort of a way to encourage that, and it's going to help us smooth the data a little bit, right? If the data contradicts that, it's going to override that prior. So again, don't want to go too technical here, but basically you can think of it as a spatially varying mixture model where you have different components for the different spot types, so the different spot types can have different expression level, and then we're going to smooth the spot types using that POTS model. Here we don't actually model the gene expression profile directly because you have, let's say 2,000, 20,000 genes that you want to model, and that would be, much too large in using these kinds of model. But what we can do is take a PCA approach where we do PCA first, and then we model, let's say, the first 15 PCs. So this allows us to compress the data into a space that's smaller, and therefore we can use sort of multivariate models or multivariate T distributions like we use here. So again, I'll focus on the results here and what we're trying to do, as opposed to really going through the model and all the statistics. But everything is part of the paper, and I'm more than happy to answer questions at the end. For estimating, we use something called Markov chain Monte Carlo, and that's pretty, straightforward or standard for these kinds of models. And then at the end of the day, what we get is all the parameter estimates. We get the spot types, we get the mean expression for the different spots, and lots of other parameters that we can explore. So I'll sort of start with some publicly available data sets. This is not cancer because there's not a lot actually of data available on cancer yet, but this is a great data set that were actually generated by colleagues at Hopkins, where they looked at basically samples from the brain and trying to understand the spatial configuration of these spots within the brain tissue. What's great is that they had done manual annotations. So they had actually provided some annotations of some of these tissues that were done manually, or based on prior knowledge on what was known about the architecture, but also based on some key marker genes that allowed them to reconstruct that structure. And I'm not going to go into the details, but basically you have these different layers that are based on gene expression of known genes. And what was good is that there's 12 samples, so we can really use that. So we can kind of use these manual annotations as ground truth and see how well are we doing in recovering the ground truth in terms of the tissue structure. Just to give you a sense of what it looks like, this is actually the expression profile of two of these marker genes that were used. This is one that defines the white matter on the left, one that defines the gray matter on the right. And you can kind of see there's much higher expression of MOB-P on the bottom corner versus, the SNAP25, which is apparently higher, has a higher expression in the gray matter. So again, everything is explained in that great paper that was published recently that I really like, because it's, it's very good from a biological point of view, and there's a lot of statistical methodologies in there as well. And all the data are publicly available, which is great. So what we wanted to do is to see how well are we doing, in reconstructing the structure of the tissue. So this is actually the best they could do in the paper using a pseudo bulk approach, where they kind of use knowledge about some of these marker genes. Then we've done sort of standard clustering, like model-based clustering and clust. And then we have two approach. One, we would do a spatial clustering-based space, where we can use the normal errors or t-distributed errors. And the t-distributed errors are kind of similar, but typically they will smooth out a lot of the outliers. So that sort of allows you to be a little bit more robust. So what we're seeing here is that we're getting pretty good concordance with the manual annotation, and certainly much better than some of the other algorithms that have been applied to this data set and the one that was published in the paper. We can actually quantify that, so we can see how well are we doing in terms of matching to the manual annotation using something called the adjusted rand index. So if the closer you are to 1, the better it is, telling you that you're really matching the manual annotations. And if you look at the results, base space or approach is on the right, and you can see that's the one that has the highest ARI on all the samples that were provided. So this shows that this does pretty well in capturing the tissue structure. And in our case, we did not use any knowledge about the marker genes. We just did PCA and applied it to PCA. Okay, so this is, not really why we wanted to apply these kinds of techniques to this technology. The real reason is because we felt like maybe being a little bit clever and thinking as statisticians, maybe we could, improve the resolution of the technology. So I told you that these technologies not single cell resolution yet. They claim it's 1 to 20 cells, 1 to 10 cells. It's probably 1 to maybe 20 and more cells, but it's close to single cell, but not truly single cell. So what we observe is what we see on the left here, right? You're going to get these spots and you're getting one expression value. But the reality is within these spots, you have a mixture of cell types. You might have, a mix of cancer cells and T cells, or a mix of different immune cells and so on and so forth, right? So you have a mix of colors. What that's going to make is that you're not actually going to really measure the expression, a pure expression of a single cell type. Is that you're going to be measuring a mixture of different cell types. So I'm going to try to explain that on the next slide, and how we're leveraging that to actually try to enhance the resolution of the technology. And I say enhance because it's not truly doing deconvolution, but it's pretty close to doing a deconvolution. So let's say, and I was just making this actually this morning. I thought the talk was at noon Pacific time. So hopefully this will be clear enough that it will be understandable by many of you. So this is actually, let's say we just have a square lattice. It's just one dimensional. You have these sort of 4 spots, these rectangles. Let's say on the left hand side you have sort of 2 T cells. Here you have 1 T cell, 1 B cell, and here you have 2 B cells, right? And that's the expression level. So because this is CD3, your T cell will be high for CD3, so they will be blue. Your B cells will be low for CD3, so they will be yellow. So this means that if you measure the expression of each spot, you will see something like that, right? Because blue is blue, and then when you have a mix of blue and yellow, you get green. And when you have just yellow, you get yellow. So you can't really know, what's in the middle, because you're seeing green. And if you look at CD3, you're like, I don't really know if it's a T cell or not, right? However, if I tell you on the left-hand side, you have something that's really blue, on the right-hand side, you have something that's really yellow, right? You're going to think that it's more likely to be like this. Probably I have one T cell that's on the left, and I have a non-T cell that's on the right. So you can kind of play this game by saying, what do I have that's next to it? And then allow you to try to deconvolve or separate the signal in the one where you have, more than cells, and the signal might be mixed. Similarly, if you measure CD19, you can sort of play that game as well, and you will know that, probably I have, B cells on the right and, T cells on the left. So by looking at multiple genes, you can actually sort of deconvolve and get a sense of what's in the different squares, right? The idea is really to leverage the spots where the signal is pure, and then try to deconvolve what you did not observe. So that's sort of what we're doing here. We try to expand the space, and I'll sort of show you that on the next slide, and then play the game as to what would it be if I know the neighbors, and then deconvolve, and then guess again, and then you iterate over all the spots. So this is basically what we call by our enhanced clustering, which is part of PaySpace. So what we do, whether we're looking at a spatial transcriptomics, a square lattice, or the Vision platform, which is the hexagons, what we do, we're going to subdivide them. So we take a hexagons and we can subdivide it here in 6 triangles. For a square, we can actually subdivide it in 9. We could actually subdivide it in 4, but we do 9 because the spatial transcriptomics is actually lower resolution. So there's probably even more cells in the spot. So that allows us to really compensate for that and increase the resolution further. Now, what we're going to do is, we're going to add some what we call in statistics latent variables or missing data. We're going to say, what if I had observed the expression profile of each of these subspots, right? We don't know what that is, of course, but we know the true expression of the entire spot. So we're going to say, we have some missing data for each of these sub-triangles that we don't know what it is. So if I look at that example here, I don't know what the expression of this, subspot here, if I just divide it in 2, I don't know what the expression here is. I don't know what the expression here is, but I can kind of play with it. I can sort of move the expression value a little bit and see whether that fits the data better, and then sort of iterate over that. And the only constraint is that the sum of the 2 things that we have here, so the constraint would be that, the blue expression and the yellow expression needs to sum up to what you observe, which is the green in this case, right? So we have a constraint. We can only move so that the sum of all the sub-expression values sum up to the true value. So again, I don't want to go into the details too much, but that's kind of the game we're going to play. We're going to enhance, we're going to add some such spots, we're going to move around to see what makes it better. Then based on that, we can estimate the spot type, and then based on the spot types, we can reiterate, and we just sort of play that game where we try to converge to something that actually fits the data pretty well in the configuration. And we can do that here because we have a spatial prior that really is looking for things that are more like one another when you have these sunspots that are touching one another. Okay, so let's go back to application. Why is this relevant, and what can we do with it? So again, we love to look at publicly available data sets here because we wanted to look at things where there were some known annotations that could be used to maybe validate some of the things we were doing. So this is one of the early data set that was published. This is actually a melanoma. This is actually a lymph node biopsy in a melanoma patient with metastasis. And this is great because we have these annotations. So this tissue was actually annotated based on the H&E stain, and they have these annotations of the lymphoid tissues, the cancer cells or the cancer, and then you have the stroma and so on and so forth. This is the older technology. So this is the square lattice. There was 293 spots that were sequenced, and then this is to remind you of the distance between these spots. So we apply base space, so our approach with 4 clusters. So you kind of have to, fit in the number of clusters that you want. You can estimate that, so you can actually look at the marginal likelihood or an estimate of the marginal likelihood to sort of see what fits the data better. In fact, here, this will tell you that probably, somewhere between 4 or 5 clusters would actually be a good selection. But because there were 4 different types here, right? There's basically the red, the yellow, the black, and everything else. We just decided to fix it to 4. So if you do that, this is sort of what you get. These are the spot types that we're finding, right? So we are finding the lymphoid tissue here. We are finding the stroma. We're actually kind of finding a mix of 2 things in the cancer. There's the cancer here and things around it, which are probably a mix of cancer and immune cells. Now, if we use the enhanced, so this is just using base space. If we use the enhanced methodology that we've built as part of it, this is what we're getting. Now, you're getting enhanced because you're subdividing each square into 9 subsquares. So it looks, much more refined. But what we're finding now is that we're finding this sort of lymphoid spot around the tumor. And if you superimpose the H&E stains, what you will see is that you're actually getting, really nice sort of lymphoid-like spots around the things that were annotated to be lymphoid tissues. So we were really happy about this because, we're using the exact same technology, but we can enhance it just using computational approaches. Now you can actually validate. So what we're finding, we can look at, expression values. So for example, we can look at T cell marker genes. We aggregated a few marker genes here, such as CD3, CD4, CD8, although some are not just T cell specific like CD4s, but it still give us, better resolution because sometimes the signal is pretty sparse. If you just look at a single gene, you might not see a lot of expression. So we aggregated a few key marker genes for these different cell types. And look at how much cleaner the enhanced version is, right? Which is on the right-hand side here versus the left-hand side. It's really, you can see some of the structure much better. And what you see, for example, if you look at the macrophages, you can see that, that sort of clusters that we found around is actually filled with macrophages because it's really high for the macrophage markers. Now we really see the lymphoid tissues, such as, T cells here and T cells around the border of the tumor as well, something that wasn't really visible if you were just to look at the spot types. Okay, this works also on, the Vision platform, and we've done that also pretty well. This is, again, another sort of publicly available data set. We've actually worked with our pathologists to enhance some of the annotations. We've added other annotations. And again, we can do the original version and the enhanced. So this is actually showing you the original spot level modeling. And we can also do the enhanced, which is the sort of shown in the Zoom version in the two things where we look at key marker genes for the basal keratinocyte and one for the myeloid cells. And if you superimpose, again, it's not as clear here because the image wasn't as good quality, or at least the annotations were very much refined, but you can kind of see that we're getting much more, discrimination of the boundaries between some of the known cell types based on the annotations. Okay, so one of the things that we wanted to see is, can we believe what we're seeing, right? Is this really real? I mean, we've had these 2 examples we've shown you where we can sort of show better match to the known annotations, but, there's no real grand truth, right? We don't know because we never measure the single cell level resolution. So what we wanted to do is to try to see if we could maybe generate in silico spatial data sets by sampling cells from single cell ionistic data. And we've actually, aggregating 6 cell types as part of the exact same data set that I told you about the squamous cell data set. They also had single cell ionistic. Here we had to aggregate those 6 different cell types because some of these cell types were too rare, so we couldn't resample a lot of cells from them. But we basically aggregated into melanocytes, myeloid cells, and epithelial cells. And then what we've done is basically sample some of these cells and then form clusters. So this is basically the ground truth that we're measuring here, and each sort of this triangle is a single cell. And then what we'd like to know is how well can we recover the ground truth. So we take these data, we aggregate at the spot level, which basically in this case will just be aggregating all of the triangles within a hexagon, and then see, can we sort of, go back to the original ground truth using an enhanced methodology. So that's sort of what we did. So this is the grand truth. This is the optimal spot level clustering. So that's the best you can do, right? If you actually aggregate and take that as a grand truth, that's the best you can do. And then this is our enhanced clustering. So what you will see is that it's not a perfect match to the grand truth because there's a lot of uncertainty, right? We're just measuring the aggregate. However, we are capturing a lot of the variability within these spots. So for example, we are finding, these sort of little triangles here that are isolated that we're seeing here, which you can think of it as sort of almost infiltrates. Same here, we're finding this, single triangle here that's on or enhanced. So this is showing you that even though it's not a perfect match to the grand truth, because there's some uncertainty, right? For example, if you have a single blue triangle, the spatial model is not going to know where that triangle is within the spot, but it's going to be able to find it. So this was really encouraging as well, because we show that it actually works pretty well. We can get pretty close to the grand truth, and we are able to get resolution that's almost as close to single cell resolution. And in fact, we can again sort of look at how well we're doing. If you look at the adjusted rand index, how well are we matching the grand truth data? This is showing you, we've done a lot of replication here, sort of simulating multiple data sets. This is, the average, so doing a box plots of all these different sort of simulation scenarios. The red line is the best you can do. So if you were just to look at the optimal spot level clustering, the best you can do by just doing, spot level estimation, you can see that the enhanced always give you, much better ROI compared to doing the spot level modeling. So this is again showing that we are able to recover some of the, almost single cell-like resolution variation within the spots. Okay, so the last thing I want to show you before, I'll wrap up and take questions is, okay, I told you about Merkel cell carcinoma. This is early on because we're doing that as part of something we'll call the Fred Hutch Innovation Lab, which allows us to partner with biotech and technology companies like 10x Genomics. And so what we wanted to do is to go back to that original sort of motivation and understand variation within our tumor biopsies, taken from some of our MCC patients and volunteers for some of our trials. So this is actually, very, early data, because we actually ran into some issues with the quality of the tissues that we had. The data actually were pretty good, but it's just the starting material wasn't super great quality. So we've generated a lot more data now that, we're actually currently analyzing. But this is to show you, one sort of tumor biopsy, again, MCC. You can see that there's some annotation. The red is inflammation immune. There's some stroma in yellow, and then the rest of it is mostly cancer. So we've done the spot level clustering. That's what you get. This time we estimated the number of clusters based on, again, something that I've mentioned earlier, the marginal likelihood. And then what we've done, the enhanced clustering, again, where you can find these, tumor infiltrates here and over here that would be missed if you were just to do the spot level clustering. So again, this is another example of something that we're able to do, find the, the close to single cell resolution data. And if we look at the key marker genes, we can again find that, we're getting much better resolution on the enhanced space. So if you look at these, the T cells, so you can see, higher expression for these T cell markers in the sort of T cell infiltrates that we're able to uncover. Same for the macrophages, we're getting much higher expression than we were getting looking at the original data. So again, this really enhances the resolution of the spot level clustering, but also looking at the expression values that we're able to sort of deconvolve through this methodology. Okay, so I'll stop here because I want to make sure there's enough time for questions. There's tons of people that were involved in some of these work. I just highlighted the key contributors. Edward Zhao is actually a PhD student in biostat at UW, who's really been the main driver of the spatial modeling. Matt Stone is a data scientist who's actually at the interface of the innovation lab and my lab. So he's working both with the experimentalists and people in my lab. He's been great, phenomenal, and getting more data, doing a lot of the processing. We have phenomenal collaborators. I highlight again Jason Bilas, who's been sort of working on the technology side of things. Paul Niem, who's our who's our MCC expert, and Thomas Pulliam, who's actually a fellow in Paul Niem's lab, who's done some of the annotation. And this wouldn't be possible without the great funding that we're getting and the partnership with Illumina and 10x Genomics. So I'll stop here, and I'm more than happy to answer questions. Thanks.
Thank you. That was great. So everybody who wants to ask a question, please use the Q&A rather than the chat. And also, before I forget, if you want to keep track of all the talks that we have in this series, the website is on the chat. It's dfhcc.harvard.edu/connectscience. All right, so I'm going to wait to see if anybody has any questions. In the meantime, I'll ask you a couple of questions. So can you go back to page 44?
This?
Yeah, so that blue triangle is not the same.
Yeah.
I just want to understand how, I don't have any intuition for how it's possible that you can find something blue there. Is it because there's somehow that, what is it? Is that like a hexagon, I guess? Is kind of blue, but only comes out as a little bit blue, so you assign it to that section?
Right, okay, so let's imagine there's that, blue triangle here, right? What's happening is when you look at the spot level data, you're averaging all these things, right? So you're basically averaging, sort of green, blue, and yellow within that, right? Or whatever is within the hexagon. And so, and when you model that at the spot level, it's going to be like, I don't know, it's more likely to be green because it looks more like a green expression profile, but it's not really green, right? And so when you subdivide it, you can actually spread the expression profile across the subspots. And so you can find a configuration where, because you have all the blue stuff, you're able to actually estimate the blue pretty well on the right-hand side. So you have a lot of information what a blue triangle looks like. And so you're able to, by moving around a little bit in the expression space, recover that, because you will say, hey, that makes more sense to actually have a mix of these different colors.
Yes, I get that. So you basically have, instead, it's like a weight of a mixture. But yeah. I guess what I don't understand is how could you possibly know which of the triangles to assign it to, or is it just randomly picking one?
Yeah, so it's yeah, that's right. So it is randomly picking one, right? It's going to move around the first one that's there, and ideally, these things can be pretty sticky because once we're in a state, it's kind of hard to move around. But in a way, in this configuration, what it will do, will probably pick the one that gives us more green that's next to one another, because remember the sort of the spatial prior wants to smooth things as much as possible, right? So it's going to try to move the triangle where maybe there's more yellow, more green right next to one another, right?
Yeah, I get that in other places. It's just that one has nothing around to get a
and that's why I'm saying, when there's only a single blue triangle, there's not a lot of information as to where that triangle should be. So it's going to put it somewhere, right?
Got it.
And once it's there, but the reality is, yeah, it could be there, but it could be bottom, it could be maybe on the, it could be anywhere within the hexagon in a way, right? Yeah. But we can't really, the data is, there's not information in the data to tell you where exactly.
Okay, interesting. So another question here. So when you showed the disease burden plot, you don't have to go back. This was at the beginning.
Yeah.
What can you say what that is exactly? What's the disease burden? And then the second question is, I'm curious, this is a very technical, in the weeds question, so I hope you don't mind. How do you do that? How do you get that data? Because I know from working in this area, it's almost impossible, it seems, to retrieve that kind of information from a patient. So you kind of get death or none. That's about it. Yeah.
So yeah, disease burden. I mean, again, you have to think about the fact that this is Merkel cell carcinoma. It's a skin cancer, so it's very easy to quantify, right? It would be, and I don't know for a fact how it's defined, but my best guess is that it's a matter of how big the tumor is and how many tumors you have, right? Because things can spread, and so you might have multiple tumors on the skin, and so you can kind of measure that and get an estimate of the disease burden. So it's kind of a function of presumably how many tumors you have and the size of the tumors.
Okay.
And so, and that might be specific to these cancer type, but we do get disease burden in many of our clinical trials. And maybe that's something that's specific to immunotherapy. I mean, I don't know.
So you get a table already with that information included. You're not having to access medical records and extract it.
We don't. Typically, that's, because we're deep, in the clinical studies, we get access to these data, which are, shared.
I see. That's from the clinical
investigators. Yeah.
That's from the clinical trial. Okay. Yeah. Okay. That's good. Yeah. I see. Interesting. Okay. Another question. So in this approach where you use correlation between things that are close, how you deal, or are you concerned about cases where I've studied data like this, that it's brain and we have very sharp edges. So if you have a sharp edge, it seems like this approach of assuming correlation would over smooth. What do you think of these situations?
No, that's a very good point. I mean, the simulation tell you that it's not really over smoothing because, for example, the blue triangle is still there, right? So we actually able to capture that. It's a prior, right? It's helping to smooth things out. There might be cases where maybe it's not, necessarily reasonable, and maybe you're not getting, super good. It's not really representing the biology, and we know for sure it's not representing the biology because biology is very complex, but it's a way to smooth and hopefully remove a lot of the noise. I don't think we have a lot of good data to really understand whether there are cases where it's not doing what it's supposed to. However, I would say the enhanced way will actually help with what you're saying. If you have sharp edges, it actually kind of help you with that as well, and that's what I've shown you in some of the slide. If you go to this is exactly what it's doing, right? There's some very sharp edges. Actually, if you look into the image, I have to find the example here where it's very sharp, right? And if you look at the enhanced, you can actually see that it's sharper around it. And so it allows you to kind of separate things a lot more, right? Which is sort of, there's an approach that's been using for image processing. That's typically what you're getting. You're getting enhanced sort of boundary resolution. So that will help a little bit with that. And you can, and obviously there's one thing that I sort of glossed over is that there's a key parameter in the POTS model that tells you how much smoothing you're doing, right? And that parameter is fixed. We fix it because it's actually very difficult to estimate because the normalizing constant depends on the parameter. But to make a long story short, you can kind of fix the parameter and see what the effect of the smoothing on the parameter. And we fixed it to a value of 3, which seems to be working pretty well across the board, at least on the data sets we've looked at. But typically it's done sort of visually to get the amount of smoothing that you need.
Another question, how does FAUST compare to Citrus?
Yeah, so that's a good question. So Citrus, it's different, right? So Citrus is really built to do predictions. So what they're doing is you do clustering, they do, I believe, sort of hierarchical clustering, and then you sort of you correlate each of your cluster, and then they do a lot. So it's really a way to identify cells that correlate with the clinical response. FAST is not really meant to do that, although we do it, but our goal is to identify all the key cellular phenotypes that we identify in a data set. And so the main difference is that it does not really depend on the the clinical response, because we just want to find phenotypes that are important, and then we're going to correlate these phenotypes. So FAST is different because it allows you to find phenotypes and actually tells you exactly what those are. So I told you these strings, right? These are CD4 positive T cells that express PD-1, CD28, HLA-DR. So you know exactly what those are. You don't need to then look at the data and see is this high for this or low for this, and then sort of do almost a manual gating. So it's a bit different because we really care about defining sort of cellular phenotypes in an unbiased manner.
Great, thanks. All right, well, one last question. Did you say this was, I'm sorry if you showed this and I missed it, but did you show if this work is published, this last part, or if it's on bioRxiv?
Yeah, the last part is on bioRxiv, so we just submitted it. So we'd love to hear your feedback. So if anybody wants to try it and things, we're still actively working on it on a new data set, and more than happy to discuss it with anybody.
Okay, so you can, if there's any chance you can put on the chat, that would be great.
Yeah, I'll do that.
Otherwise, we'll just look for it. I mean,
Yeah.
it can't be hard to find.
Yeah, I mean, if you Google base space and bioarchive, that'll come up.
Okay, good.
But I will put it on the chat as well.
All right, well, I'll give people maybe another 30 seconds to ask a question. I've already answered all the ones that were asked, I think. Okay. Well, and if nobody asked, then I'll thank Rafael one more time. That's a great talk. I will look forward to learning more about what you talked about today. And I'm going to send you a preprint too.
Sounds good.
All right. Thank you, everybody. Bye-bye.
Thanks, everyone.